Okay, well, thank you very much. When I have conducted projects over my career, I have really tried to emphasize interdisciplinarity and I've tried to emphasize connections between different kinds of research areas. And actually, as I look at my title, it doesn't look very interdisciplinary, so I decided to add, especially for a gut microbiome meeting, I at least wanted to add some indication that I will be trying to provide a broad approach, and I'll be integrating it with ideas about gut microbiome biology. So really what I'm going to try to do is provide one sort of theme. And given that my background is in evolutionary biology and studies of infectious diseases, the theme I'm going to try to present is that when we broaden approaches to try to include different perspectives, often we'll see assumptions that might be limiting the kind of potential for new advancements. And I think that's true I think we'll see that's true if we try to integrate these areas I've just talked about, sort of evolutionary biology and studies of infectious disease and studies of gut microbiome biology. And just to sort of give a sort of a tangible sense of where I want to go, I've just given this little illustration of one sort of model of way of looking at the relationship between gut microbiome changes and disease. And I'm going to suggest that when I look at this, there's something that I find a little bit concerning. And I'm going to focus on that thing that I'm going to find concerning, try to develop it from different perspectives and see whether or not we can generate an insight that might be useful for us in a practical way. And the point that I find concerning is it pertains to this middle box, that the mechanism by which microbiome alterations in the gut may influence disease is by leading to an aberrant growth of commensals and abnormal effect of those commensals, which then brings about the disease. But rather than directly talk about what it is that I find concerning with that idea, I want to try to kind of build up an approach to disease from scratch and then fairly quickly come to this idea of whether we may need to modify our sense of the interaction between microbiomes and commensals as a mechanism by which disease is brought about. So I like to start, just to make sure I'm not leaving anything out, I like to start out with an approach that sort of includes everything. And the reason I like this is as we learn more and more about chronic diseases, we find, sort of going back to this discussion that we just had, that if you think just about one thing or another thing, you're missing something. And so you have to actually think about many different things interacting, and it really does, I think, become quite complicated. And so as a way of kind of starting out from a base where you think you're kind of including everything, I like to think about this triad of disease causation in which we can envision any disease being a consequence of contributions from the individual's genetic makeup, from influences of the environment, sort of the non-infectious environment or non-symbiotic environment, and then also interactions from the symbiotic environment. And then we can say, well, how do these things fit together? If I'm just looking at one thing, am I maybe missing something, a big part of the picture that could be useful? And at the outset, I'm afraid because these terms are used in such different ways, I want to tell you how I, as a biologist, would define these terms. And I think a very useful way that's becoming more and more standard in biology is to think of symbiosis or symbioses as the entire spectrum of intimate associations from parasitism through mutualism. Okay, so if we're thinking about the symbiotic corner, we'd be thinking about intimate agents, living agents that are ameliorating in their effects on disease or protective, and then agents that are actually damaging. And again, we need terms, and the terms often are used in different ways, so I want to take a minute to define what I mean by terms like parasitism. So I'm defining parasitism very broadly, which is the way I think things are moving now in science. And so I would define it as an association in which an individual The parasite lives in or on another individual, which we call the host, and causes harm to the host. Slipping down to mutualism, I would define mutualism as association that provides fitness benefits to both interacting individuals. So here we're bringing the evolutionary approach in. We're saying it's very hard to define maybe parasitism, mutualism, or commensalism unless we're thinking in some way of some measure, some unit. And so that's one of the advantages of thinking from an evolutionary point of view, is that we evolved because of differences in evolutionary fitness, and so we can think about harm or help to a host individual in terms of fitness units. So if the fitness unit is, the net fitness is negative, then we call the association parasitism. If the net fitness is positive, we call it a mutualism. And then it sort of brings us back to commensalism. And generally, wherever you look, in biology at least, commensalism is defined as an association in which an individual lives in or on a host individual without harming or helping the other individual. And I think it's sort of this point that we need to kind of come back to integrate our sense of how we categorize different organisms and how microbiome alterations might influence health. Before I come back to that commensalism, though, I want to just then state what I mean when I talk about infection. And this infection is defined very differently in medical circles and other circles. But I just mean infection as a state of parasitism. Okay, so if we come back to commensalism, if we think of commensalism in terms of some objective unit of net benefit or net harm, I think we come to the conclusion that commensalism really, if we could measure all the various effects of an interactant on a host, we have to say that commensalism is really a boundary line between parasitism and mutualism on a continuum, which we can call the symbiosis continuum, which ranges from obligate mutualisms, in which a host is dependent on a mutualist. So we think of lichens, for example, as an obligate mutualism, or we can think of original eukaryotic cells as resulting from mutualism between a mitochondria, a bacterium, and a host, which eventually became mitochondrium and a host. And then it goes through commensalism, which is where you just have the net benefit, the net cost or net benefit to the host being zero, and then all the way to mild parasitisms and lethal parasitisms. And that spectrum has become much more important, I think, over the last 10 or 20 years, as we've realized the failings of thinking in health science circles about lethal infectious diseases. I think now pretty much everybody's thinking about this in terms of an evolutionary perspective, comes to the conclusion that we could have anything under the sun. We could have natural selection leading to very intricate, detailed, virtually perfect adaptations of pathogens and have those, or parasites, and have those parasites be lethal, 100% lethal, or anywhere in between. And so as soon as we recognize that an association is a parasitism, then we kind of have to ask, how harmful is it? And how does that influence our understanding of the role of that parasite in any problem, whether it's dysregulation of a microbiome or the disease that's associated with that dysregulation? So that's sort of where I'm going right now. And just to begin with, I'm focused on inflammatory bowel disease just because we want to keep our focus on something that's manageable in 35 minutes. So just to sort of begin with, I want to talk about one organism that's been strongly associated. It's been, let's say, significantly associated with inflammatory bowel disease, and that's adherent invasive Escherichia coli. And often when that association is discussed, and it's believed that this is playing a important role in at least some cases of inflammatory bowel disease, when that association is discussed in microbiome circles that are dealing with inflammatory bowel disease, they're referred to as commensals, but as we understood them better, we realized they've got definite virulence factors that could cause harm to the host. And so people are a little bit of a loss. They weren't, well, maybe it's not right to call them commensals, so now people started calling them pathobionts. But I would just say that a lot of this it reminds me a little bit of the epicycles and Ptolemaic astronomy where you sort of come up with something and you say, oh, well, it's not quite right. Let's sort of invent a new term and think of a new process. And then we can kind of explain the motions of the planets in the sky. So I would say that this is kind of leading us in the wrong direction. I would just say we just come back to the idea that probably if we could measure everything precisely, there isn't really going to be anything that's truly commensal. So this is a parasite. And whether it's a mild or a harmful parasite depends on a lot of things, a lot of environmental things. So very well is going to depend on the microbiome. But when we think of it that way, because we know that parasites can really mess up a host, then it opens another kind of dimension for looking at IBD. And so for thinking of IBD, we know that anytime you've got parasitic contributors to a disease, that there's everything we study indicates that there are genetic vulnerabilities to that. So there's going to be a genetic influence. And we know that with IBD that there are associations with environmental microbiome environmental influences on microbiome composition. And so those environmental influences we can talk about there. And we can talk about the components of the biome that might be protective, and we could put that in as in terms of a mutualistic aspect in the symbiotic vertex. But generally we'd say, well, IBD is probably somewhere down here in this triangle. We put it down here to reflect the sense that the microbial interactions are important, but also environmental interactions may be important. We really wouldn't know where to put it there, but recognizing that adherent invasive E. coli is really parasitic, it should make us cautious about thinking that the direction just goes in terms of environmental disruption of biome, then leading to a vulnerability to this parasite, because we know that the parasites could actually have an effect in the opposite direction. So this is sort of a cautionary point that is based on incomplete knowledge that pertains to our interpretation of microbiome changes associated with IBD. What I want to do now is sort of shift to another microbe that now is pretty much being accepted as playing a contributing role in inflammatory bowel disease and other serious gastrointestinal diseases that has been looked at quite a lot from a point of view of microbiome disruption, and that's Clostridium difficile. So I would say in the last 5 years, pretty much everybody, most of the people at least that are looking at Clostridium difficile in inflammatory bowel disease are saying, yes, it's playing a role in some, maybe pretty many cases of inflammatory bowel disease. And almost everybody says Clostridium difficile is a pathogen or microbial parasite. And so we don't have to be concerned in this case about commensalism, although a lot of people were sort of thinking of it as a commensal gone wrong that was just causing problems, probably pretty long time ago, just causing problems when you had antibiotic treatment in a hospital and people that were vulnerable. Then you had clostridial overgrowth, and then you had severe consequences of that, largely because it's resistant to antibiotic treatment and it's associated with antibiotic treatment. And so the general sort of sense that has developed is that it's a result of the antibiotic treatment, and I'm certain that that's true to a substantial extent. But again, coming back to our discussion, just because you understand that one influence is occurring doesn't mean that one should ignore what may be a much more complicated situation. We've got a lot of interactions between different contributing factors. And so what I want to do is just probe a little bit more deeply into what some of those possible interactions could be and how they may shed light on the role of Clostridium difficile and what we might be able to do about severe clostridial diseases, Clostridium difficile diseases, whether they be classified as inflammatory bowel disease or other kinds of gastrointestinal disruptions. So just to kind of summarize what some recent reviews have said about Clostridium difficile as it relates to severe gastrointestinal disease and the microbiome, particularly with regard to how you could intervene, maybe to control clostridial disease, it's really hard to get a generalization that you can say this absolutely is trustworthy. So I've sort of written down some of the points that various experts have made when they're trying to review the literature. And so these are their opinions. They look pretty good, pretty balanced, but I'm just presenting them as their opinions. So in terms of safety and efficacy of fecal transplants in uncomplicated Clostridium difficile infection, the sense is that it's pretty good for moderate infections and for recurrent infections. It actually tends to resolve things pretty well. When we're dealing with more severe infections that just occur at the moment, or also severe recurrent infections, the sense is it's not so clear. It might be providing some benefit, but it's just not as compelling as the benefit that seems to be documented for moderate recurrent infections. And then we go to inflammatory bowel disease, particularly, we look specifically at ulcerative colitis and Crohn's disease. It's much more uncertain, and at least Kump and Huckenauer said there's no reliable support for fecal transplants improving Crohn's disease condition, and then some promising results for ulcerative colitis, but maybe even some negative results for IBD in terms of flares. Okay, so one sort of general way to think about this current state of affairs is it looks like if C. difficile infections are sort of moderate, then altering the microbiome can actually help. It doesn't really tell you whether the alteration of the microbiome caused the problem in the first place, but it's consistent with that idea. So what I want to do is now come back to sort of the evolutionary argument and say, well, given that we pretty much all agree that Clostridium difficile is a pathogen, then it becomes important for us to understand what is the basis for the variation in the harmfulness of the association of that pathogen with the host. Why sometimes are people carrying C. difficile and not getting any symptoms at all? It's actually quite common. And then sometimes people in a life-threatening situation, and not infrequently, they die from C. difficile infection. So what I want to do is sort of come back to now an evolutionary approach and say, given that Clostridium difficile is a hospital-acquired infection, at least it's really severe mainly when it's associated with hospitals, what do we know about severity of hospital-acquired infections from an evolutionary point of view? And one of the general themes that's come out of evolutionary approaches to the virulence of infectious disease organisms is that if the infectious agent can be readily transmissible from very sick individuals, then natural selection will favor the evolution of a high level of harmfulness for those pathogens. It's really quite logical. It just says if somebody's sick and they can't transmit the pathogen, any variants of the pathogen that cause people to be sick aren't going to get transmitted, okay? And so they're not going to be making up the future population. However, if you have the potential for transmission of pathogens from very sick individuals, any pathogen that exploits the host sort of ruthlessly and causes that host to be sick and can replicate in that process and produce a lot of progeny to get transmitted to new hosts can get away with it. Okay, so in hospital environments, there's a situation that allows these pathogens to get away with exploiting the host very intensely. And that I would refer to as just attendant-borne transmission as a way of having a term to use. And so this is just diagrammed in this little picture. So we have an infected patient and a pathogen with a really highly exploitative variant of any pathogen. Because we know these hospital-acquired pathogens are largely transmitted on the hands of attendants, at least the ones that are causing big problems in hospitals. And we know a lot about the details of that transmission. We also know that that transmission can occur from an individual that can't move out of bed, can't even move. Let's say if a diarrheal disease, that individual is releasing millions, even billions of organisms, and that release can contaminate the environment, and we know that that then contaminates hands, and then attendants move from one patient to another, and if they're not careful about using gloves or washing hands, then they can transmit the pathogen to other hosts. And this is how these hospital-acquired infections just cause terrible problems in hospitals. And most of these pathogens that are responsible are really good at surviving on the external surfaces that might be touched by hands, or on the hands themselves, or on the gloves themselves. And this can be blocked very easily by just instituting good hand washing guidelines. The problem is that it's very easy to have those guidelines turn out to be sort of too much trouble. So you see in hospital environments, you get lapses in these procedures that we know can prevent this kind of transmission. But from an evolutionary point of view, this suggests that when we get this transmission in hospitals, we're going to cause evolutionary changes in these organisms in the hospitals that are going to make them much more damaging. And this is an alternative explanation to what people generally, how people generally explain the damage associated with hospital-acquired infections, which is that, people in the hospital are very vulnerable, and it's true. And so a given organism that might not be very harmful outside of that setting can be lethal inside the hospital. So that certainly is part of the answer, but just because it's part of the answer doesn't mean that there's something else a little bit more sinister going on. And so many years ago, after I proposed this idea, I provided the first test of it, and the test just looked at all E. coli outbreaks in hospitals in the US and the UK that occurred before we realized that we could treat them with antibiotics. And so I just looked at how long the outbreak lasted, and I looked at the deaths per infection. So this is E. coli, which we normally think of as being not all that harmful, but you can see sometimes it was causing 10% lethality in these hospital nurseries. But the really intriguing thing is if you look at the duration of outbreak and deaths per infection as a function of that, again, before antibiotic treatment was available, you see there's this strong association. Each data point corresponds to an individual outbreak, except for when we get down below, in these short durations, we had to add outbreaks together to get a meaningful percentage until you got a minimum of 10 infections. Because what's happening is people would recognize we're getting some baby deaths, got to figure this out. Typically, some hospitals decide we better close down the ward, clean it up. Other hospitals would say, well, let's keep going. And then those other hospitals ended up having a long duration of outbreak. So this is kind of significant that you're getting these high lethality rates that are significantly associated with the duration of the outbreak. Okay, so now that is sort of a little bit of a background. So how does that then relate to Clostridium difficile? So we know that Clostridium difficile is mainly a problem in hospitals. It's been explained as a problem in hospitals because of the vulnerability and also because of the high use of antibiotics in hospitals, which would then tend to kill off the more vulnerable organisms and then leave Clostridium difficile, which is inherently more antibiotic resistant. And we know that it's associated with microbiome alterations, and we know that those restorations can improve the situation. And that then suggests that the microbiome is kind of important. But because we also know that Clostridium difficile is a pathogen, it's not just a commensal, but it's really a pathogen that can have very severe effects. It then still brings us back to that question that maybe the pathogen could be causing some of this disruption itself, Clostridium difficile. And so even if the microbiome restoration, as indicated by the fecal transplant results, is inhibiting C. difficile, it doesn't mean that it was the alteration in the microbiome that opened the door for C. difficile, which is what you might expect if it was just a commensal. It could very well be that C. difficile is competing with these other organisms, causing organisms to be knocked out if the C. difficile is particularly virulent, and then we can counter that by reintroducing those competing organisms. Okay, so it could be essentially that the C. difficile is responsible for some of that microbiome alteration, even though the restoration of the microbiome, to some extent at least with fecal transplants, is protective. It's actually helpful, at least in moderately severe C. difficile infections. So what this then, I think, leaves us is with a recognition of the need to understand what's really going on with C. difficile in hospitals. If the E. coli data represents a reasonable explanation, then we could go to look in the literature and try to find out, is or do experiments or studies to try to find out whether the C. difficile in hospitals is inherently more harmful than the C. difficile we get outside of hospitals, or does it look like it has the same level of harmfulness and it's just increased vulnerability, increased antibiotic usage in hospitals. And I want to emphasize it's not either or. It's very likely all of those things would be playing a role, even if the C. difficile in hospitals is more harmful than the C. difficile outside, if it's actually evolved to be a more nasty pathogen. And so what, one of the things one of my students and I have been involved with is looking in literature and trying to sort of figure out what does the literature suggest. The literature suggests that it's more nasty in hospitals. It's actually evolving to be more harmful in hospitals. And so I just want to quickly go through some of that evidence. It's sort of stepwise kind of argument. So you can look to see, first of all, that hospital-associated C. difficile is associated with greater mortality. 15 in one study in Örebro, Sweden, 15% of the hospital-associated were cases ended in death. 4% of community-associated did, whether you adjust for concomitant disease or not. So adjusting for concomitant disease is kind of important because it deals at least a little bit with this idea that people in hospitals may be more vulnerable. But it only deals with it a little bit. There are a lot of potential alternative interpretations of this data. And one of them is that, of course, it doesn't control for the vulnerability in hospitals all that well. But another is that if it's community associated, it could still be hospital associated. People come out of the hospital sometimes with the C. difficile that they get from the hospital, and then they may come down with the illness, which would be, potentially a hospital strain. And so we really sort of need to try to assess that contribution before we can understand what community-associated mortality figures really look like. But at least it's consistent with the idea that that C. difficile in the outside community is less harmful. Another study from New York addresses this to some extent. And in that study, they looked at the individuals who had a community onset, and then they asked, did this individual actually have contact with somebody that we knew had a Clostridium difficile inside of a hospital, within a 1 month period. And so they subdivided the community onset between a community-acquired C. difficile, that's people who didn't have any contact with a C. difficile infected individual in a hospital, and those that did have the contact. And these results really are quite interesting to me, at least. If you look at deaths per case, 3.5% death per case for hospital-acquired, and almost the same for community onset that could be traced to hospital. So these people that left hospital weren't sick at all, but then they became sick outside. Maybe people just came in for some other purpose or visited a person in the hospital who had Clostridium infection. And then no deaths in the community acquired. And then if you look at other kinds of characteristics, they also accord with the community acquired being less harmful. Whether the individuals, when they finally generated a GI tract problem, whether they actually stayed, whether they had to have hospitalization or not, or how long they stayed in the hospital, and whether they had a recurrence. Okay, that's not applicable for these because they were in the hospital to begin with. But so overall, it's consistent with the idea that the problem is worse in the outside community. And it also deals a little bit with the health issue because these people are not people who had underlying health problems that would have made them more vulnerable. But it also is not complete in a number of ways, but especially because they were looking at people who came down who had symptoms. And it could be in the outside community, you have a whole lot more people who are carrying chlamydia, I also mentioned chlamydia, Clostridium difficile, who never even would show up in a study like this because they never have any symptoms. And so one way of dealing with that is to look at the ratio of asymptomatic infections to symptomatic infections in the outside community and also in hospital long-term care settings. And so we can calculate the percentage of infections that are identified by surveys in the outside community and the percentage, and calculate the percentage of those infections that are symptomatic. And so we also did that. You can use In this case, in Sweden, there's some really good data. So we used 3 different studies from Sweden. It allowed us to calculate that in the general population, using the overall rate of asymptomatic infections and the overall rate of symptomatic infections, about 99.5% of the infections are asymptomatic. Whereas if you look at long-term care facilities like hospitals and nursing homes, 83% were asymptomatic. So still a high portion of, even in these settings, high proportion of people were asymptomatic. But we know that it was a pathogen. It was just an asymptomatic infection with the pathogen, and a highly significant difference. So this gives us a sense, another sort of piece of the puzzle that suggests that these hospital-acquired organisms probably are more nasty. And as people have looked at the genetics of the community-acquired and the hospital-acquired organisms, I think the picture becomes clearer. For a long time, people were just studying in hospitals, and so you didn't have that kind of comparison. But even though people weren't looking very carefully in hospitals, outside of hospitals, they were identifying the strains in hospitals, saying these are just a few strains that are just rip-roaring through hospitals and traveling around the world. Then when people finally looked at strains outside of the hospitals, they realized those hospital strains are there to some extent, but they're not the predominant ones. They actually tend to be quite rare. And so people that have been looking at this, not really from the point of view of natural selection and evolution, but rather from the point of view of, sort of bacteriology and, mutations and infectious disease analyses, they're concluding there's something kind of interesting going on here. And so here's a quote from Volker, and just 7 years ago. It said, during the past decade, this is a time when it really became aware that Clostridium difficile was causing problems. During the past decade, mutations transformed C. difficile from a rare nosocomial infection to one that can spread rapidly in hospitals and has spilled out into the outside community. Now infections of healthy individuals with fewer known risk factors is not uncommon. Okay, so this sort of gets back to, can we explain it all just in terms of antibiotic treatment or not? And increasing recurrence rates have been reported. Okay, but so one point I want to sort of emphasize is they're talking about mutations. They're not really talking about natural selection. So they're not talking about that argument that I was raising that in a hospital environment you actually select for the nastier strains. They're just making observations saying, we've got these genetically distinct strains. And typically it was studied in terms of ribotyping. We've got these genetically distinct strains that are causing real havoc in hospitals. And if you sort of merge that with the data that I talked about before, we know that some of those ribotypes, some of those strains, genetically distinct strains, spill out in the community and cause problems in the community. But if you look at the strains that are not hospital associated, they're causing almost no problem at all. They're asymptomatic to the percentage of infections that are asymptomatic is extremely high, as I was showing you before. Okay, so this sort of adds this other dimension. Remember I was saying what I try to do is try to integrate ideas from different perspectives, different bodies of knowledge, and hopefully come up with some insights, sort of something to add to a problem that's mainly being looked at from one particular perspective. This just summarizes what I was saying in a citation, that they're rarely isolated in the general community. And so if we sort of wrap this all up in terms of Clostridium difficile, we can say that there's an implication of all of this, that we're getting evolution of increased virulence in hospitals, but if we intervene in a certain way that removes that competitive favoring of the nasty strains in hospitals, we should be able to get mild strains. We should be able to favor mild strains. And then if we link that back to what we were looking at before with regard to fecal transplants, remember it was the sort of the more moderate Clostridium difficile infections that seem to really be responsive to the restoring of the microbiome. So that then suggests that maybe, now we look at this more in terms of evolution of virulence of pathogens, recognizing some of these pathogens that are involved in some of these microbiome-associated illnesses could be altering their harmfulness and therefore could be altering the way in which they interact with the host and the microbiome. Then we can sort of reconstruct that triad of disease causation and say that there is another aspect of non-parasitic environmental risk factors, and that would be the behavior attendants, which then can alter the organisms that we would put in the parasitic category that could be disrupting things, and then as a result of the reduced microbiome or the compromised microbiome makeup, then they're able to actually cause really damaging infections. But if we could actually remove this, then what we'd be doing is making C. difficile infections more mild, and then we know that the microbiome restoration tends to be more effective. At least that's what the data suggests, that once it gets to the point that you've got massive problems, you've got perforations of the colon in, say, Crohn's disease, it may just be too late to do that. But if you actually can avoid the problem in part in the first place by not having C. difficile having such negative effects, as soon as you get the more mild problems surfacing, then the restoration, restoration may work better. And so, this is sort of another way of looking at it, sort of that sort of harks back to the original figure that I gave, which is to say that once we understand that there are these infections that can be responding to selective pressures in the environment, if we alter them, then, and then also that if we realize that the infections could be altering, this is going both directions, infections could be altering the microbiome and vice versa, then we actually might have a much more manageable problem. And then the last point that I'd really like to make on this brings us back to how pervasive this problem is around the world and in different socioeconomic strata. And the answer is it's all over. Okay, it's in wealthy hospitals, it's in hospitals that don't have very many access to resources, and what's known in general is if people really get conscientious about improving hygienic standards in hospitals, their hospital-acquired infection rates drop precipitously. And that's not just a little deal in terms of public health. In the United States, there are about 100,000 deaths from hospital-acquired infections every year. And so, just by comparison, there's 500,000 or 600,000 deaths from cancer, a little bit more than that, deaths from heart attacks and stroke. This is more than twice as many deaths from hospital-acquired infections in America than deaths from automobile accidents or guns. And so we're dealing with a big issue, and it looks like even at an epidemiological time scale, if you block that attendant-borne transmission, you really can make big inroads, and it might be that you're going to get even bigger inroads once you take into account the evolutionary effects. And then finally, sort of to relate this to this idea of socioeconomic status, these interventions can be done even with hospitals without resources. So this is a kind of intervention that is, it's almost unforgivable not to do in hospitals with a lot of resources. But studies have been done, and they say, half of nurses don't follow appropriate hand washing guidelines, and about 70% of doctors don't. But you can flip that around if you intervene. But really importantly, I think that these are interventions that can be done in hospital environments, long-term care facilities that don't have access to resources. It just doesn't cost much money. It just costs a little bit of time. And then you get paid back by having a better resolution of these problems. So anyhow, on that sort of happy note, on a very sort of concerning problem, I think I'll leave it and open the floor for questions. Thanks.
Yeah, so that's great. So you're showing us that you can tame a parasite, basically, and make it less virulent and make it open to treatment. Have you, going back to your continuum, right, from obligate mutualism all the way down to lethal parasitism, do we have examples of the things going the other direction, right, where you have obligate mutualists, where you get an imbalance and where you start seeing the evolution of a parasite, more of a parasitic relationship, and what is it that would drive that?
Right, okay. That's an interesting question. I've actually looked at it a little bit differently. For me, it's easy to see why we would have parasitism. It's harder to sort of picture why we have mutualism. So I've always said, well, so when would you go from parasitism to mutualism? Okay, and so I think it probably goes the other way too. I just actually haven't thought about it. Maybe we can just put what I'm about to say in reverse, and then we can get it. So basically what I was saying is if you have a symbiont, living inside of a cell, let's go to the early eukaryotic cell. If you have a symbiont living inside of a cell, and it can do something better than that cell can do, then it's at least partially mutualistic. If you've got a bacterium that's good at providing energy that's better than the bacterium it's living in, let's say archaeobacteria and a eubacterium living inside. So then it's at least partially beneficial. But then if you have any change that has altered that ability of that bacterium that's living inside the bigger cell to be horizontally transmitted, then its interest is entirely in line with the interest of the host cell. And then, well, even if that organism was partly parasitic and generating more energy, so it was, let's say, there, but still partly mutualistic because he's maybe giving a little bit of energy to the host. Once transmission is only vertical, let's say, and it can happen just by a simple mutation, then natural selection favors it to be completely mutualistic, favors it to go to a mitochondrion, okay? Because the interest of that endosymbiont then is the same as the host. Okay, so that's one way. That could be partly related to mutualists in the gut, but I think that probably the way in which mutualists in the gut is related to this continuum is more in terms of understanding the conflict of interest between the host and all these other organisms are genetically different, but realizing that those organisms can provide benefits to the host in terms of protection against other organisms, because they're competing with invading organisms. They could provide vitamins and so on, other nutrients. And recognizing that it's, in some ways, that conflict of interest, you would think, might just always favor exploitation by those microbes, because they can evolve very quickly, and what's to keep them from just exploiting? Well, part of what's to keep them is how natural selection's acted on the host, because the host is kind of in control in a lot of ways of this situation. If things go well, those microbes are, making, good defenses against other invading microbes or providing nutrients. The host is our physiology, our immunology, presumably would be selected to just say, fine, let it just go away. As soon as something goes wrong, I'm going to change the game here. I like to sort of envision it. I like to compare it to people who are sort of bad sports in Scrabble games, because my sister-in-law was not a very good sport, and when she was losing the Scrabble game, she tipped the game up and turned it over, okay? And that marriage didn't last that long, maybe for that reason, but for me it was very useful because it provided sort of a sense of what we have as the host with a lot of physiology and immunology to work in our favor, what we can do. If the situation isn't going right, we turn the Scrabble game upside down and start over. We do it by diarrhea, basically. We wipe it out, say, okay, now I'm going to recolonize. And maybe recolonize from the appendix, other places. So we have the control to say, if this microbe makeup is working in our favor, great. If it's not, everybody loses, everybody except for me, as long as I can survive the diarrhea. And then we start over again, and then what you eventually should be doing is selecting for microbes that don't try to push the boundary. There'll always be that tension. There'll always be mutations for microbes that say, I'm going to get a little bit more out of this. I'm not saying it, obviously, but they'll be selected to get a little bit more out of it. But if they do, then they can end up losing because the host is controlling it. And I think that probably is the answer for most of these mutualistic associations in which you pick the mutualists up from the microbe rather than handing them down vertically. Okay, so that's my long explanation. Sorry, it was long. So the question is, how about the other way around? Well, I've sort of covered that a little bit. The selection pressure is always there for mutualists to cheat, to get a little bit more. They're providing the vitamin, or whatever, a little bit of protection against invading organisms. What if they provide a little bit less? What if they exploit a little bit more? They don't provide that vitamin, but they use it more for their own benefit, and then they sort of flip over a little bit into the parasitism end of it, and presumably it's happening all the time. But we just have our immune systems and our physiology flexible enough that we can say, game over, tip the board upside down, and we start over. And so I think that's probably what's going on. But I would think it's almost always going to be the case that these things are flipping over here, and probably more rarely flipping over there. It's harder to explain that sort of mutualistic association, but we see it. We see it with sea organisms that fluoresce. You know, they're picking up bacteria that allow them to fluoresce.
Teeny follow-up, because I'm sure there's other questions, but would you propose or hypothesize that when you have vertical transmission from mother to offspring, that you basically need to get somebody all the way to reproductive age, right?
So
there's a sort of,
that's right.
It's like,
That's a trick. And there may be a lot of these things because we know that you do get that kind of colonization, but there's a lot of turnover, okay? So, and we also know that if you don't have that selection for mildness that would result from following through succeeding generations, we have some of the nastiest effects on babies from pathogens that that they're vertically transmitted for one generation. I mean, the STDs that are really sexually transmitted, just devastating for babies, causing blindness, all sorts of terrible problems. So it's a tricky thing to be thinking about because you have to take into account the vulnerability of the babies and the pathogens that are actually evolving to take advantage of us, and then they get in these vulnerable babies that got blindness and all sorts of problems like that.
I happen to like pantomime, but what do you call C. diff pathogen when your data showed, using your figure, that it is probably commensal, maybe benign parasite? It is a potential to be pantomime because 95%.
Okay, so that sort of gets at this definition again, and I would just say that we can't use percent asymptomatic infection as evidence for commensalism, because almost, if you look at almost all the pathogens that we understand well in terms of the proportion of infections as symptomatic, almost all of them cause a lot of asymptomatic infections. Even, the L-tor Vibrio cholerae organism, which can cause terrible infections, half the time it's asymptomatic. So I think we have to understand that when we're dealing with infectious diseases, that we're, as a rule, going to have a lot of asymptomatic infections. And so that's why I try to come back to a way in which we can distinguish what we mean by commensalism from what we mean by infection. And I would say that we have to sort of average over all of the associations. They have 1,000 people who have that organism and say, what is the average? Is the average net negative? And so even though you could pick out, let's say with cholera, you could pick out 50, or Clostridium, you could pick out 50 individuals that didn't have any apparent harm. We know these organisms have characteristics that will cause harm. It may not be apparent, and we know that they can cause terrible harm. So overall, we still call them a parasite if we're being objective about it. And so really what I'm saying is I'm suggesting there should be sort of a change in semantics. But if people really like the term commensal, I would be happy to say, okay, well, let's just think about what we mean by commensal, which is that sometimes it's damaging and a lot of times it's not. But then it becomes kind of a little bit of a term that's a little vague and ambiguous.
Even in cholera or salmonella or Shigella, I agree with you that some of them have no symptoms. But if you look at the physiology of the gut, there is deleterious effect.
Yeah.
And that has not been shown in
I know.
severe.
Yeah, you're right. So we need to look. But I think, but even with some Vibrio cholerae, for example, it's different with the invasive organisms like Shigella. But with Vibrio cholerae, which is not invasive, you could look and say, well, I don't see any gut pathology for these mild infections. So do we call it a commensal or do we call it
in those, the AMG is upregulated and so on.
That's right. Yeah. So that's okay. I'm happy with that progression of these ideas. And I would say, well, then let's look and see what's going on in those quote asymptomatic infections. We may find some pathology there.
I love you with that.
Okay.
Many of the questions you had in my mind, I think I can reconcile with that figure.
Yeah. I think we can reconcile really a lot of, it's really...
About this figure, because you talk about it the whole time, and it's a great figure, but it's completely simplistic. Because there's no such thing as an axis from good to evil. It's extremely multidimensional, and everybody knows this, of course. Yeah. And it depends on the environment, it depends on this, and sometimes some things that can be beneficial in the short term can be detrimental in the long term. Yeah. So this sort of axis of evil depiction is nice to sort of introduce your talk with, but it's not good to actually really want to.
I would say it's really good. Just to agree with you on that, because it doesn't say that these other things aren't having an effect. It just says that we need to have something objective if we're going to be defining terms in a way that's not going to be ambiguous. Okay, so, you know, you
mean ambiguous, because this thing is ambiguous.
no, I think it's saying you just look at the net fitness effect in any given population. I'm sort of, well, okay, but I'm going to get to where you are, okay, in just a second, okay. But I'm just saying you look at the net fitness effect in any population, and then you say in that population, it's going to be a parasite. Okay, it's going to be a parasite or it's going to be a mutualist. And then you can say, well, there are these other factors going on. There's time scales, there's other, you know, there's nutrition, there's also the other things I had in my triangle. Okay, right. So then what we can say is, well, let's think about our sense of parasitism and mutualism in a way that could be context dependent. Okay, and that's not ambiguous. That's just saying, let's make sure it goes back to the triangle. Let's make sure that when we call something a parasite or a mutualist, that we're saying just for this context, just for this population, just for this interval. And you know, so even for a mosquito, you know, with a malaria organism, you could say there's a certain amount of time that malaria organism is actually a mutualist for the mosquito. When it's in the host and it's causing that host to have a fever and causing the host to be delirious, the mosquito can get a good blood meal. Once a mosquito takes the malaria organism, it doesn't cause too much harm, but it's causing a little harm, and it can cause big harm by blocking the feeding apparatus when it goes to feed its next host, which means that it has to feed for longer and get swatted. So then it is acting in a parasitic way. So I'm fine with all the other influences you're talking about. I'm just saying that we'd have to apply that spectrum in a context-specific way. Yeah. Okay, I guess I took two, so I cheated a little bit, but okay, thanks.